This video will finish up Chapter 6 on enzymes. I'm going to show you a couple examples of how enzymes work to perform their function. And we're going to start with the enzyme chymotrypsin. During digestion, proteins must be broken down into smaller peptides, and this is done by proteases. And chymotrypsin is an example of a protease. And in particular, it's going to cut the peptide at specific locations. The structure of chymotrypsin shown here consists of three chains, which are labeled the A chain, the B chain, and the C chain. You can see that the B chain has some antiparallel beta sheets. The C chain has an alpha helix. Disulfide bonds are shown in yellow. D here shows a close-up of the active site of the enzyme bound to the substrate. The aromatic part of the substrate is fitting into the hydrophobic pocket. The hydroxyl group on the serine 195 residue will attack the carbonyl group of the substrate, and then the developing negative charge on the oxygen is stabilized by what's called the oxyanion hole, where the amide nitrogens from the serine 195 residue and the glycine 193 residue stabilize it. Now I'm going to go through the sequential steps of the chymotrypsin mechanism. So in step 1, the substrate will bind to the enzyme, and the aromatic side chain will fit into a hydrophobic pocket on the enzyme, positioning the peptide bond for attack. So here is the substrate, and it's going to fit into the enzyme, and notice that side chain is fitting into the hydrophobic pocket. And notice here that I'm showing the serine residue 195. That's going to be important in the next step. Nucleophilic attack occurs in the next step. Interaction of the serine 195 and histidine 57 generates a strongly nucleophilic alkoxide ion on the serine residue, and this attacks the peptide carbonyl group, forming a tetrahedral intermediate. So here, the oxygen is attacking the carbonyl carbon, breaking that pi bond and moving those electrons up onto the oxygen. The negative charge on the oxygen, shown here, is stabilized in the oxyanion hole through hydrogen bonding to the NH groups on the serine 195 and the glycine 193. So here we see the hydrogen bonds forming, and this is a stabilizing effect. In the third step, that tetrahedral intermediate will collapse. So one of the lone pairs on the oxygens comes back down to reform the C double bond O, and now we're going to break the CN bond as shown here. So this is how we're cleaving that peptide bond. So we're going to release part of the substrate, and then we end up with this acyl enzyme intermediate. Interestingly, the histidine residue, histidine 57, facilitates this group leaving with its proton. In the next step, water comes in, where it's deprotonated by the histidine 57 residue to generate a strongly nucleophilic hydroxide ion. And that intermediate is short-lived, so just like we saw before, it will collapse. So a lone pair on the oxygen comes back down to reform the C double bond O, and now we kick off the enzyme, essentially. Then the hydroxide ion will attack the carbonyl, again generating a tetrahedral intermediate where the negative charge on the oxygen is stabilized in the oxyanion hole. In this step, the tetrahedral intermediate collapses. So a lone pair on the negatively charged oxygen comes down and reforms the carbonyl and breaks this bond here, releasing the substrate from the enzyme. And in the final step, the product dissociates from the enzyme. So I'm going to show you the animation here for that mechanism. So now the substrate is going to enter into the active site of the enzyme. Notice how the benzyl group here is situated in the hydrophobic pocket, and there is a hydrogen bond stabilizing the carbonyl of the substrate. That hydrogen bond is formed between the carbonyl oxygen and the hydrogen attached to the nitrogen of the serine 195 residue. Now moving along, we see that the hydroxyl group attached to the serine 195 attacks the carbonyl, and now we've formed our tetrahedral intermediate where there's a negative charge on that oxygen, which is in the oxyanion hole, stabilized by hydrogen bonds to those two groups. And in addition, there is a hydrogen bond between the histidine 57 residue and this nitrogen. Now the next thing that's going to happen is that tetrahedral intermediate collapses, cleaving the peptide bond. So now we see cleavage of the substrate, and part of the substrate is leaving and part is still left behind. So now water is going to come in, and water is going to be deprotonated by that histidine residue, and that's going to facilitate it attacking the carbonyl of the substrate that's still there. And now we have a new tetrahedral intermediate, which then collapses, removing the substrate from the enzyme, and now it's free to leave. Now that you have seen the mechanism for how chymotrypsin works, how would you classify the enzyme? I know I said it was a protease, but that would be a subclass. Which of the 6 classes would that fit into? An oxidoreductase, a transferase, a hydrolase, a lyase, an isomerase, or ligase? Think about it and discuss it in the chat. An understanding of protease mechanisms like the chymotrypsin mechanism led to the development of HIV treatments. Only 3 key enzymes operate in the HIV life cycle, and one of them is a protease. So this is a figure showing how the HIV protease can cleave a peptide bond. Drugs have been developed as HIV treatments, which are HIV protease inhibitors. So what they do is they form non-covalent complexes with the HIV protease enzyme, and they bind it so tightly that they can really be considered irreversible inhibitors. And it prevents the HIV protease from doing what it needs to do. And these are some of the chemical structures of the HIV protease inhibitors that are currently used as drugs. Another enzyme I want to talk about is lysozyme, which is an enzyme that cleaves peptidoglycan, which is a polysaccharide found in bacterial cell walls. And we haven't talked about polysaccharides yet. We'll actually get to that in the next chapter. And cleavage of the cell wall leads to lysis of the bacteria. So this shows a peptidoglycan polymer, and in particular, the lysozyme will cleave that CO bond. So with the assistance of water, lysozyme will break this polymer into 2 segments. The mechanism occurs via 2 successive SN2 steps. An aspartate residue acts as a nucleophile that attacks a carbon in the first SN2 step, and then glutamate 35 acts as a general acid protonating the leaving group. Water hydrolyzes the intermediate, and then glutamate 35 acts as a base to deprotonate water in the second step. So originally it was proposed that this proceeded through an SN1 mechanism, but more current evidence correlates more to an SN2 mechanism. So I'm going to focus on the SN2 mechanism. So here, the residues that are important on the enzyme are shown. So the aspartate residue performs an SN2 attack. So it's the nucleophile attacking this carbon, resulting in cleavage of this CO bond. And the proton on the glutamate 35 facilitates the leaving group in leaving. So this part of the molecule leaves, but you're still left with this covalent intermediate. So in a second step, glutamate will deprotonate a molecule of water, facilitating it attacking that same carbon kicking off the aspartate residue connected to the enzyme. And now you have your second product that's now freed from the enzyme, and it's ready to dissociate and leave. The biosynthesis of peptidoglycan involves an enzyme called transpeptidase, which links two chains together, and it's facilitated by a serine residue. So here's the transpeptidase, and it is facilitating linking the two chains together to form a cross-linked peptidoglycan. The way penicillin works is that it interferes with the ability of the transpeptidase to perform its function. So this is a picture of penicillin here, and the transpeptidase, in particular the hydroxyl group of the serine residue, attacks the penicillin instead of performing its normal function, and then when it does that, a derivative is formed which is inactive. Now, interestingly, bacteria, in response to our introduction of penicillin, developed an enzyme called lactamase, which inactivates penicillin. And the way those work is that they attack the penicillin and make the penicillin inactive. So just like the penicillin made the transpeptidase inactive, the bacteria fought back, creating a different enzyme to make the penicillin inactive. And then how did scientists respond? They developed compounds such as clavulanic acid, which is termed a suicide inactivator, which then irreversibly inactivates the beta-lactamases. So here this shows the clavulanic acid and how it interacts with the beta-lactamase, resulting in an inactive beta-lactamase. The last topic is the regulation of enzyme activity. And this can occur via non-covalent or covalent modification, and it can be reversible or irreversible. Let's start with non-covalent modification, also referred to as allosteric regulation. Now, allosteric effectors or modulators are generally small chemicals, and they can be positive, meaning they improve enzymatic catalysis, or they can be negative, meaning they reduce enzymatic catalysis. Now, typically, in many allosteric enzymes, the substrate binding site and the modulator binding site are on different subunits of the enzyme. And we can call the catalytic site the C site and the regulatory site the R site. So here, if we have a positive modulator, it could fit into the regulatory site, which makes the catalytic site want to bind the substrate more. Let's exemplify this by looking at an enzyme called aspartate transcarbamylase. It catalyzes the conversion of aspartate to N-carbamyl aspartate. Here we have the enzyme, and it can be in either the inactive T state or the active R state. The catalytic sites are shown in shades of blue and purple, and the regulatory sites are shown in shades of beige and yellow. When the molecule CTP is bound, it favors the inactive state, but when CTP is released, it favors the active R state. CTP stands for cytidine triphosphate, and it is an allosteric regulator, and it is an essential nucleotide. So in particular, CTP is a negative regulator. When it's bound, it favors the inactive state. The kinetics for allosteric regulators differs from Michaelis-Menten kinetics and looks more like what we saw for the hemoglobin saturation curve. So the curve is sigmoidal or S-shaped, and this indicates a transition from the low activity state to the high activity state. And then this graph just shows several different plots which represent the effects of several different concentrations of a positive modulator plus or a negative modulator minus on an allosteric enzyme. You can also get regulation through reversible covalent modifications, and there are over 500 different types of covalent modifications that have been found, and here are some examples. Phosphorylation in particular is a very common one. So in this, you're adding a phosphoryl group to an enzyme. Another would be methylation. So in this, you're adding a methyl group, a CH3 group, to an enzyme to modify it. So, a particular example of an enzyme that can be regulated by covalent modification, in particular phosphorylation, is the enzyme glycogen phosphorylase. And that enzyme catalyzes the reaction of glycogen plus a phosphoryl group to form glucose 1 phosphate and the shortened glycogen chain. The enzyme has two forms called the alpha form and the beta form. When the phosphoryl groups are covalently bound to the enzyme, it's in the phosphorylase alpha form, and when they are not bound, it's in the phosphorylase beta form. The more active form is this alpha form, and the less active form is the beta form. The addition of the phosphoryl groups is mediated by another enzyme called phosphorylase kinase, and then the removal of those same groups is mediated by another enzyme called phosphoprotein phosphatase 1. So notice how we're getting more complex here, and there are a variety of enzymes working together. For some enzymes, an inactive precursor called a zymogen is cleaved to form the active enzyme. And chymotrypsin is an example of that. So in that, it starts out as chymotrypsinogen, which is inactive, and then trypsin comes in and cleaves the chymotrypsinogen into two segments. It cleaves between the residues 15 and 16, and then the residues 14, 15, 147, and 148 are removed to get to the active alpha chymotrypsin form. Similarly, the enzyme trypsin starts out as an inactive trypsinogen, which is then cleaved to form the active trypsin enzyme. Now, some enzymes use multiple types of regulation. The activity of glycogen phosphorylase, which I was talking about, which is used in muscle, is subjected to a multi-level system of regulation shown here. So this part indicates what I had on a previous slide, where you can add phosphoryl groups or remove them to alternate between the active and less active forms of glycogen phosphorylase. And that's mediated by these enzymes phosphorylase kinase that adds the phosphoryl groups, and this phosphatase phosphorylase, abbreviated PP1, that removes the phosphoryl groups. Now, what this complex map is showing you is that these enzymes are also further regulated by the hormones insulin and glucagon. So there can be quite a sophisticated map and sequence of regulation for enzymes. In summary, in this chapter, we learned why nature needs enzyme catalysis, how these enzymes accelerate chemical reactions, and in particular, we looked at chymotrypsin and a lysozyme. We looked at kinetic studies involving Michaelis-Menten kinetics. We talked about enzyme inhibitors, competitive, uncompetitive, and mixed inhibitors. And then we talked about the regulation of enzyme activity.